My name is Lauren Gottschalk, and today I'm going to be discussing RNA and its catalytic functions. So life requires a lot of things, but one thing that it requires molecules that have the ability to catalyze. So the question is, would RNA do this? Would it fit this agenda? So to answer this question, I'm going to answer it the best I can by offering evidence from my lecture class, my textbook, and a scholarly article. RNA, or ribonucleic acid, is a usually single-stranded molecule that transfers genetic information, just like DNA does, but RNA differs from DNA in its base composition and slightly in its molecular structure. So RNA is going to have 2' OH groups, and one of the base pairs, adenine, is going to pair with uracil instead of thymine, which it will pair with thymine in DNA. RNA's usually single-strand composition should not be viewed as a disadvantage. In fact, scientists believe RNA may have been here before DNA. It's not believed that RNA was exactly first, but it is believed it was here before DNA. So some precursors likely would have been polymers similar to RNA, but simpler, and they could have evolved to pave the way for RNA. So in an RNA world, it is hypothesized that RNA was the primary transmission of the genetic material and only stepped back when DNA arrived and became the primary transmitter. DNA is much more stable than RNA. RNA is very vulnerable to hydrolysis, and DNA lacks that OH group, so it's not going to worry about degradation. So it's only natural that DNA would take over as the more stable molecule. So RNA doesn't just disappear. That's not all it does. It's still the main player in the synthesis of proteins. In 1982, Thomas Scheck discovered catalytic functions of RNA. So this was a huge discovery. At this time, a lot of people didn't accept that as a fact. But further research has proven this to be true. At least 9 catalytic RNAs have been studied and described, and more are being created in the lab. So RNA catalysts are called ribozymes, and most ribozymes function by positioning metal ions at their active sites. In this slide here, I've just got a simple example at the top of a ribozyme, how it would be cleaving or cutting the RNA. So that's an example of the ribozyme there. And then below it, I've got the ribosome is a ribozyme. And this diagram just very briefly explains that ribosomal proteins act to orient the catalytic RNA. And so this is important because RNA can change its orientation to perfectly line up for intended reactions. So this is really neat to think that it can orient itself to know exactly what to do and where to go and how to act as a catalyst. So catalytic functions are super important for RNA metabolism and processing. Proteins are synthesized by catalytic RNA, and in the lab, RNA can catalyze reactions for their own reproduction, actually. So another thing that can happen with RNA that's pretty cool with enzymatic activity is it forms a stem loop structure, and it is also called a hairpin loop just because of its appearance. So the strands actually fold back upon themselves, which allows interactions between the loops. So this can create more complex structures. Here we have what is known as the hammerhead ribozyme. This is one of the most understood and studied examples of an RNA catalyst. So it was identified by William Scott, and this ribozyme is involved in replication in some viruses. It works by catalyzing cleavage of one of its own phosphodiester bonds. So Mr. William Scott determined through X-ray that this hammerhead ribozyme forms 3 alpha helices, and the nucleotides of the stems form normal base pairs, but the loops interact with a variety of other reactions. The only thing about this one, in a way, you can't treat it as a true catalyst because it's its own substrate and it can't undo its conformation to return to the original conformation. It also doesn't use metal ions as most enzymes do. However, there are still plenty of other ribozymes that are considered true catalysts. So a lot of the ribozymes that we know about so far are ones that have been created in the lab. And so based on experiments with them, scientists have actually kind of explored that it seems like the main difference between protein enzymes and ribozymes is simply the speed of reaction, not the reaction type itself, which is super cool because this is more evidence supporting that maybe there was an RNA world. Maybe RNA really was here before DNA and was fine in the early aspects of life, and just, got demoted when DNA, which was a better molecule, came through. So in my conclusion, based on the data I presented, it does provide some evidence that RNA was here before DNA, and a lot of scientists believe in this RNA world. So I attached this little picture just because I thought it was kind of neat. But this is just the idea, so I'm going to leave you with this, and you think about it and see what you think. And thank you very much.